Howdy. So this is actually lecture 7, molecular biology. So I know it's going to sound kind of intimidating, but it is really interesting. It's like how we get from this molecule of DNA to the people that we are. Like, how does this one little molecule dictate everything that your cells do and look like and become? It's really interesting. So this comes from the chapter 9, from Concepts of Biology. So hopefully by the end of this lecture, you will be able to describe the structure of DNA, explain the process of DNA replication. So when cells need to divide, like mitosis or meiosis, you have to make a copy of the DNA. So how does that actually happen? So we'll talk about that today. So hopefully by the end, you can explain the process of DNA replication, explain the importance of this enzyme called telomerase to DNA replication, explain what we call the central dogma of molecular biology, and that, again, sounds so random out of context, but by the end, you should hopefully know what it is. What is transcription? How do you make proteins? How do ribosomes make proteins? Describe the genetic code and how the sequence of nucleotides determines what a protein will be and do, and why not every cell expresses all of its genes. So for introduction, right now I feel like we mostly, in normal life hear about DNA when it comes to things like crime solving, paternity testing, human identification, and like genetic testing. So in daily life, we probably associate, just the word DNA with some facets of these things, like the DNA at a crime scene being matched to a suspect. Paternity testing, like in Jerry Springer or Maury, like the DNA test determines you are the father, and then they like run off screaming. Human identification, so when like crimes happen and there's like a body found, how do we determine who that person is if we have no other clues? Genetic testing, so like 23andMe and Ancestry, DNA tests. So right now we kind of, mostly associate DNA with all these things, but DNA is relevant to all of our lives every day, all the time. And so when we think about DNA, like where can we find it? It's in all your cells. So when you're doing like forensic science, DNA can be retrieved from like hair, but not the hair itself. Like there's no DNA in this. This is just protein. But if you were to get like a hair that happens to have some follicles still from the scalp, that's where the cell would come in. So DNA can be retrieved from hair follicles, from blood, so the red blood cells, or saliva. And so everybody's DNA is different. And with the exception of identical twins, everybody's DNA is unique, and it's possible to detect differences between human beings, but also similarities between families. So this is probably old now, but for a little bit there was this kind of like viral thing, this notebook that was like, if I go missing. And so people would put samples of their DNA. That way if they ever go missing, they have, like a reference for that. But for some reason they're like, okay, let's do nail clippings. But nail clippings on their own, like nail also is just protein, it's just keratin. And so your nails aren't going to have any DNA on the clippings. Like they might have some underneath. If you've been like scratching your skin or stuff, you might release some cells and have them underneath your nails. But like the nails themselves don't have DNA. The hair itself doesn't have DNA. Sure, they could kind of make a comparison between like a hair structure, but again, there's no DNA in your hair. It's all in like the scalp. So you would have to like pluck your hairs, and enough that you're pretty sure that there's like the little white spot at the end that would have your DNA in those cells. Yeah, so if you're gonna do that, at least like get cells that actually have DNA, not just random stuff. So that's, yeah, kind of to help show like, well, where is your DNA? It's in your cells, like your actual cells. So another thing that we kind of like think about when it comes to DNA is Dolly the sheep. So this was the first cloned mammal it was an identical copy. They were able to take the DNA that coded for this one sheep, and then they put it into a different cell, and when that cell started dividing and becoming like a full-grown adult, it expressed the same things, so that's what made it an exact clone of the other one that they got the DNA from. So that's the cloning process. Like, there's complicated, ways that they do this, but it's basically taking the DNA that codes for something else and putting it in cells that will then express that and become a clone of that same organism. So how did we get to know what we know about DNA? So in the 1950s, well, first of all, we started talking about Mendel, right? So Mendel did all these experiments, and he was like, okay, something gets passed on from parents to offspring that pass on their traits, right? So like a flower is always gonna give rise to another flower, and it's gonna look similar to the parents. So something is passed on. So scientists over time had to like figure out all the pieces to this puzzle. So what is it that gets passed on? So they figured out that it was DNA, but at the time they knew what like DNA was, but they didn't know exactly the structure. So in the 1950s, these two guys, Francis Crick and James Watson, worked together to help like determine the structure of DNA. So if you are studying a molecule that's so important, it codes for like everything for an organism, you want to understand its structure. So that's what they started doing. They're trying to figure out like, what does this molecule actually look like, and how does it work? So there were other scientists also working on this, like Linus Pauling and Maurice Wilkins. And so on this other team in the Wilkins lab, there was a researcher named Rosalind Franklin, and she was using X-ray crystallography to help understand the structure of DNA. So this method, like, you prep a molecule, like some kind of substance molecule, and you prep it a certain way and you put it in this machine, and the machine shoots X-rays at it, and depending on the structure of the molecule, it like reflects X-rays back in a certain pattern based on the structure of the molecule. And so this is what this grad student, Rosalind Franklin, was doing. She was like, prepping the molecule and trying to see what it would reflect like so that she could determine the structure. And so on the right is this picture of her X-ray pattern, like the X-rays, how they bounced off the DNA molecule. And it might not make a lot of sense to us, but her looking at the way that the X-rays bounced off helped her figure out, like, okay, so it must have bounced off this specific way because this molecule is this structure. And so she was the one that basically helped realize that it's a DNA double helix like we picture now. And so actually, it's Rosalind Franklin that took this X-ray picture, the same basically one picture before, and on the picture on the right, that's Rosalind Franklin. So she's the one that took this picture, and basically Maurice Wilkins stole it, took her notebook, and like conferred and sent the information to this other team, Francis Crick and James Watson, and they were like, aha, so that's the structure. Okay, so they started like writing it up and publishing it, and so they're the ones that get credit for discovering the structure of DNA, even though it was her picture that, like, without that they would not have been able to do that. So this is James Watson and whatever Crick. So yeah, these guys were awarded a Nobel Prize for basically getting that work published fastest. And unfortunately, like, she passed away in the meantime because she was working with X-rays, and at the time, there wasn't a lot of knowledge and protection, as much as we have now, about X-rays and cancer, and so she developed cancer and passed away. So they would not give her a award afterwards, because they're like, oh, sorry, we don't give Nobel Prizes to the deceased or whatever. So I always like to give her a shout out, because she actually, you know, without her picture, they wouldn't know the structure of DNA. Maybe eventually somebody else would have done something like that, but like the way it played out, she helped discover the structure of DNA as we know it today. Yeah, so unfortunately, science is great and its findings help humanity, but it's not always equitable. And yeah, that's just kind of the reality of it. So hopefully it's going in a better direction now, but science sucks sometimes. So now we know more about the structure of DNA. So DNA, we picture, you know, that molecule like a ladder that's kind of twisty. But now we know that it's made of smaller pieces. So the pieces are called nucleotides. And so each nucleotide has 3 parts. So there is a sugar, so a 5-carbon sugar, 1, 2, 3, 4, 5. Each point is a carbon, represents a carbon. So a 5-carbon sugar, a phosphate group, and a nitrogenous base. And so those 3 things make up this one molecule that we call a nucleotide. And the base could be one of 4 different options. So this is what we call the backbone, these other 2 pieces, the phosphate and the sugar. And then the base could be one of these 4 things on the right. So it could be this other group called cytosine, which we represent with a C, thymine, which we represent with a T, guanine, which we represent with a G, and then adenine, which we represent with an A. So they look kind of similar at first glance, the structure and the shape looks very similar, but the smallest differences in chemistry make a big difference, and so that's why they're different molecules with different names. And so, like I said, one nucleotide is all 3 pieces, but this piece, the base, could be either one of these 4 things, C, T, G, or A. Those are the 4 nucleotides that make up DNA. And so one of these are like building blocks, and so you link one with the next one to the next one to build the overall DNA molecule. So yeah, that's the chemical structure. And here is another picture kind of showing that just a little bit more simply. So if we think about the ladder, DNA actually has 2 strands that are connected to each other. So one strand has, one nucleotide that's connected to the next nucleotide that's connected to the next one. And here we can show, a simpler representation with just the letters. And then there is the backbone represented by just a line. And so one strand connects with the opposite strand because of those bases that are there. So the ones that are in a certain position can only bond with a certain other one because of the way that the molecule is, and the small little details and compatibilities of atoms in chemistry. So A and T always bond together because they just happen to have, those complementary things sticking out. And then G and C always bond together, again, because those particular groups that stick out in that molecule are compatible. And so that's how the 2 different strands kind of stay together. It's because these strands are bonded across from each other by hydrogen bonds. And so that's what keeps a double-stranded helix together. So here is another picture showing that you don't have to know the structure, but just to show what I mean. So thymine and adenine have these 2 chemical groups that stick out and can reach each other and are compatible, and so they bond with each other like that. So A and T can bond with each other. And then guanine and cytosine have these groups that stick out and can reach and like are compatible, and so they bond together. And so it's always T bonds with A and G bonds with C. And so one strand we call the 5' strand, or one end. So that's because if we count the carbons, 1, 2, 3, 4, 5, there's this group sticking out at the 5' end, and then this end has a different group sticking out at this end, and we refer to that as the 3' end because this sugar is 1 to the 3rd sugar. So this is probably more detail than you need to know, but just to show you, like, okay, there's a lot more to the molecule that we know about now and how it works. And so DNA is the same in all organisms. Like, all DNA molecules are made of those same 4 nucleotides, and they have the same 4 bases. So adenine, guanine, thymine, cytosine. No matter what living organism it is, it expresses, you know, it has the same molecule of DNA. Like, if you were to extract its cells and rip out its DNA, it would look, the molecule might look like pretty similar to yours. So the only thing that's different is the sequence of each organism and like the organization. So if I, for example, were to take the DNA in one organism and put it in a totally different kind of organism, technically that new organism could express that DNA because it's the same thing it knows. It's like the same DNA molecule. It's just giving it a new recipe. Yeah, so hopefully that makes sense as we go. So all living things use these molecules to build their DNA. And it's just the sequence that's different. So RNA is also a nucleic acid, and it's similar. It's really similar. There's just, again, tiny, minute differences that make it a different molecule. So DNA, you know, is made of nucleotides, and so is RNA. So RNA also has a 5-carbon sugar, but the tiniest difference makes this a different sugar. So in DNA here on the right, it uses deoxyribose. So if we look at this carbon right here, or the group sticking out from this carbon right here, so deoxyribose just has an H, a hydrogen. And so in RNA, it uses this sugar called ribose that has an oxygen here. So it's OH. And again, overall, the structure is really similar, but the tiniest difference makes a difference. And so that's why this is ribose, because it's kind of like the normal version. And then this is deoxyribose, because now there's no oxygen here at that position. So they both use nucleotides, but the nucleotides that build RNA have this oxygen here. So they use ribose instead of deoxyribose. And that's why it's RNA ribonucleic acid, and DNA is deoxyribose, deoxyribonucleic acid. But the nucleotides are, you know, also built from a phosphate group that sticks out over here, and then a nitrogenous base that sticks out over here. And most of the bases are the same, so A, C, G. And then the only thing that's different is that RNA uses uracil, and that we represent with a U, instead of the T. So RNA will use U instead of T. And those slight differences, like that different molecule, the uracil, and then the ribose, help RNA actually be single-stranded. So RNA is just single-stranded, but DNA is always single or double-stranded. So how is DNA arranged in the cell? You might have remembered from the cell tour that DNA is in the nucleus. So that's because DNA is so important. It's like the recipe book for like everything that the cell needs to do and make and be and like become. And so DNA is always going to be kept in the nucleus. So DNA has 2 main jobs. To get copied when a cell is ready to divide, by either mitosis or meiosis. And it must be read to produce the molecules like proteins that carry out like the stuff of the cell. So people call it like the instruction manual. I kind of refer to it as like a cookbook. So my sister gave me this big cookbook that's like this thick, and it tells you how to make like everything. It's called, I actually don't remember what it's called. Shows you how much I use it. But so it kind of tells you how to make everything. It's like not just recipes, it's also like how to debone a fish, how to do this from scratch, how to make pasta, and that's relevant for all the other stuff. And so yeah, like I said, that book is this thick. And so I refer to DNA like that. Like it's a huge cookbook that tells you basically everything you need to know. And because it tells you everything, like it should be kept pretty safe. And so that's why DNA is always kept in the nucleus and doesn't come out of the cell. Or out of the nucleus, unless the cell is getting ready to divide and the nucleus, dissolves. So DNA is really important, and so it's protected and packaged in very specific ways. And DNA is actually super long, so if you were to stretch it out, it's actually really, really long. And so it has to be like wrapped and organized specifically so that it's kept safe inside the nucleus. So here's the double helix that we think of, but it's pretty long, and so we kind of have to like wrap it around proteins called histones, and then you wrap them tighter and tighter, and then you can coil those and wrap it tighter and tighter. So kind of like Christmas lights. You don't really want them to just be like a long, stringy, loose mess. Like you can coil them, and then you could kind of like make that, a smaller and more organized structure so that it's like safe and less chaotic. So that's how DNA is actually organized. We refer to DNA, and then we also refer to chromosomes, but what's the link? The DNA is wrapped around stuff tightly until it's like condensed into a chromosome. So when a cell divides, the cell has to prepare, if you remember, by going through interphase. And it's during the S phase of interphase that you make more DNA. So S stands for synthesis, or like making something. So S phase is when you synthesize DNA. And so the actual process that we make DNA is called DNA replication. And we'll talk about how you make more DNA. So because the two strands are what we call complementary, meaning like, the bases on one strand pair up specifically with the base on another strand, we can kind of use one side to help build the other side when we need to make a new molecule. So basically one DNA molecule is like a zipper, and so you can unzip it and use each one to make the other half of the zipper, if that makes sense. So here is like the gray showing like the parent molecule of DNA. You unzip it, you use one strand to build another half, and then the opposite strand also builds another half, so that by the end, now you have two DNA molecules. Whereas you started with one, now you have two because you unzipped it and used each half to build the rest. So that's what we call the semi-conservative model of replication. It conserves some of like the parent strand, but you make the other half new. So here's another version of that, kind of just showing you unzip the DNA, and based on what's there, what letter is there, you can put the next letter. So if you know A and T always go together, if there's an A here, then you'll put a T. If there's a C here, G and C always go together, so no matter which orientation, like if there's a C, then there's going to be a G. If there's a G, it's going to be a C. And that's how your cell reads this half and then builds the other half of the new molecule. So by the end, you have two identical strands of DNA. And so this is overall the big picture concept that's important to, kind of remember, that when you make new DNA, you're basing it off of the original DNA molecule. You just unzip it and then build more. And that's how at the end you end up with two identical molecules of DNA. And so this is the like more complex version. We don't really need to get into this, but it is more complex than that. So let's just skip through that. And so just by the mechanics of how this process happens, there happens to be some pieces of DNA that get cut off every time you make a copy. So at the very end of like the DNA strand, when you make a copy, there's a little piece that gets cut off. And then so technically every time you copy DNA, each copy is a little bit shorter and a little bit shorter and a little bit shorter. So because of that, there is this kind of like thing that evolved to happen. So these things are called telomeres. And basically there's this enzyme, telomerase, that adds just repeated kind of like fluff sequences. And so they add like kind of fluff sequences so that those will be at the very ends of the DNA. So that eventually, like over time, as the DNA gets copied and gets like a little bit shorter and shorter, it's the fluff DNA that gets cut off. Like it doesn't code for anything. It doesn't, yeah, it's kind of just like a protective layer, like a cap of extra sequence that doesn't code for anything. But when the DNA molecule gets shorter and shorter every time you make copies, at least it's just cutting off like extra bits that doesn't matter over time. So it's this enzyme, telomerase, that adds those repeat sequences at the end called telomeres that help kind of prevent, you know, the DNA, important parts of DNA from getting cut off every time there is DNA replication. And this is kind of relevant because the reading about Henrietta Lacks and her cells, so her cells have an overactive telomerase enzyme, and so they keep adding these ends, and so that is what helps her cells repeat and keep dividing in the lab endlessly, because it's not having this the telomeres get shorter and shorter, because that's what limits how much a cell can divide, how long the telomeres are. So if you have more enzyme activity of this thing, telomerase, then these cells could theoretically live forever, and that's exactly what's happening. So as long as you keep like feeding these cells in the dish the nutrients it needs, and like taking away the waste products, then those cells will keep dividing. So yeah, that's actually what, you know, the research has found about Henrietta Lacks' cells since then, to explain why they're able to divide indefinitely. And it was this scientist, Elizabeth Blackburn, who discovered how that enzyme works. And so later research, she saw this in other cells, but later research in the HeLa cells confirmed that it's telomerase, and it is present in human cells, not just whatever they were studying it in. So sometimes mistakes do happen in DNA replication. So when DNA gets copied, sometimes there are mistakes introduced. Like you'll read a letter and then put the wrong letter. So incorrect bases, usually the enzyme that puts the letters there will notice its mistake. It'll be like, you know, adding things in an assembly line and then like, oh crap, like sorry, that's the wrong one. So it'll take it out and put the correct letter, and then it can continue. So a lot of times it'll catch its own mistake. If they are not caught, then this mistake may result in what's called a mutation, which is just like a permanent change in the DNA sequence. So if it doesn't get caught, then that DNA eventually is going to get replicated, and then that mistake will just keep getting carried on. So that's how mutations like occur and then get carried on throughout that organism, and like species. Yeah. So that's how you make more DNA, but how do you make stuff from DNA? Like how is it the instruction manual? How does it code for all these different things? So that is what we refer to as the central dogma, which is basically this idea. Again, everything has to have like its own name. That DNA codes for RNA, and then RNA codes for protein, and that's how you make things. So the central dogma is really just this like kind of flow of information. DNA to RNA to protein, and that's how things get made. So genes in the DNA specify sequences of RNA, and those specify the sequences of proteins. So I kind of, again, my analogy is like a cookbook. So the DNA is like this big, huge cookbook with all the recipes you could ever need. When you need to make one recipe, you don't really need to take the whole book into the kitchen because then it could get damaged. So what is more convenient is to get like an index card or something and write down just the recipe that you're going to make at that time. So say my favorite food is lasagna, so I always use that. So say I'm going to make a lasagna. So this huge cookbook, I'll just write down like everything I need and the steps to make a lasagna, and then I can take this index card into the kitchen. And it doesn't matter if it gets destroyed, you know, it's fine, because then I can always just go back and make another little like index card version of the recipe. So that's what RNA is. So DNA is like the big cookbook, and RNA is like that index card where you're just writing that little copy of that recipe. And then you could take that out of the nucleus and actually go and cook up your protein. So then protein is like, you know, your actual meal. So say, I don't know, you're making like baked chicken or whatever. So you're cooking up protein based on the little index card recipe that you got and copied from the DNA. So that's kind of the flow of information. It's like a big cookbook that you don't want to take into the kitchen and destroy and, you know, spill stuff on and whatever. So you make a little index card version of the recipe in RNA, and then you can cook up the protein. So this is actually 2 steps. Each arrow is a different process. So this first arrow from DNA to RNA is called transcription. So transcription is you're transcribing or writing that recipe down onto the index card. You're transcribing, or like scribes, you know, medical scribes are like people that write things down. So transcription is like you're writing the little recipe on the index card. So DNA is read or, you know, written, transcribed into this molecule of RNA, and technically it's called mRNA. So it copies just that one recipe, and then the RNA can go outside the nucleus into the cytoplasm and then start cooking. So transcription is a little bit similar to DNA replication. You unzip the DNA, but just a little bit in one region of that recipe. You write down the recipe, but based on what's there. So again, it's kind of like you're building the molecule based on what you're reading on this half. So if there's a C, you put a G. If there's an A, because this is RNA, you would put a U instead of a T. And you would just go down that line, and that's how you know how to write the RNA based on what is there, what letters are there in the DNA. So there's extra detail here that you don't necessarily need to know. Like, again, this class is kind of transitioning towards becoming more about how it relates to daily life. So yeah, we can, you know, kind of skip some of this detail. But this process of transcription makes a little RNA. And so now this RNA can leave the nucleus, go into the cytoplasm, and gets translated. So translation of that mRNA kind of translates it from one kind of molecule to another kind of molecule. So it goes from being a nucleotide-based information to a protein, which is like a totally different molecule, right? So that's where translation comes into place. Like, It's actually translating into something being done. So kind of like the cooking, it's not super similar, but kind of like the cooking, like you go from it being words to now something cooked, something physical. It's like a different form. You translate it into something. I don't know if that helps. So the ribosomes are in charge of making proteins, and this is how it's taking part in this process called translation. And it's reading that recipe, and then it can tell like what ingredients it needs to put together. And in this case, the ingredients are amino acids, since the building blocks for amino acids for proteins are amino acids. So you put one amino acid building block to the next amino acid building block, and then that's how you build a protein. So ribosomes do that. They have two parts to it. They read the letters that are there in the RNA, and then they put one amino acid, and then it reads the next few letters and then knows, okay, that means that I need to put this amino acid. The next three letters says I need to put this amino acid, and so on. And so that's how it builds a protein, by reading the code, and it tells it to put this other amino acid molecule, and then it adds them together, and that's how you build a protein or a polypeptide. And so we have, figured out since what sequence of letters codes for what amino acid. So then you can kind of know, okay, if this is the sequence of DNA, I know it's going to build a protein made of this amino acid, and then this amino acid, and then this. It's actually like super impressive that we figured it out. So the ribosome reads 3 letters at a time, and those 3 letters tell the cell, okay, you're going to put this specific amino acid, or these letters is going to make this specific amino acid be there. So that's how you would kind of like be able to, even ourselves, like look at a sequence and say, okay, so the ribosome would probably build it this first, and then this one, and then this one, because the ribosome reads 3 letters at a time that we call a codon. So a codon is made of 3 nucleotides. It reads those 3 and then says, okay, I need to put this amino acid here. Okay, this 3 means I need to do this. And so that's how your cells know how to put together all the different things that it needs. So we've learned a little bit about proteins and how much they do in cells. Enzymes do a bunch of different reactions. And so proteins really are like the major chunk of what your cells need and make to like do things and communicate with each other and do all this different stuff. So that's how gene expression is. So yeah, let me go back to that beginning slide. So this idea of the central dogma is basically what we refer to when we talk about gene expression. So a gene is one recipe, and then when the protein gets built, that's how it's getting expressed. And so that is what leads to the different like phenotypes from different genotypes, because of these 2 processes of transcription and translation. And so not all genes, even though all genes have the exact same DNA, so all genes in one organism, remember, have all the chromosomes needed for that organism, but they don't express all genes at once. So for example, like a skin cell is a skin cell because it only expresses the genes it needs to be a skin cell. And that's different than like your liver cell or your nerve cell. Like each cell is very specialized. And even though they technically have the full cookbook for everything any cell in your body needs, it's only expressing a certain set of genes at a time. So for a certain cell to function properly, the necessary proteins have to be made at the right time. So not all of the genes are going to be expressed at all times in all your cells. It just would not be possible. And it would take way too much energy, and the things would not be compatible. It's just not possible. Yeah, so the process of turning a gene on to produce the RNA for that gene and express and make that protein, that's all gene expression. So cells in our super complex, multicellular organisms like us are specialized, and that's why each cell is different, because it's just expressing like genes for that kind of cell. So a muscle cell is different from a liver cell, is different from a skin cell, and that's because there's different expression of different sets of genes in each cell. Yeah, so hopefully this all makes sense now. So hopefully you can kind of describe the structure of DNA, how it's like the double-stranded molecule. Kind of roughly explain the process of DNA replication. You just like unzip one molecule of DNA and then use that to build and end up with two molecules. Explain the importance of this enzyme called telomerase to DNA replication. Explain what's called the central dogma, or gene expression is just basically taking the DNA, writing down that one recipe in the form of RNA, and then cooking up that recipe in the form of protein. Describe the different steps in protein synthesis, so basically what I just said. Describe the role of ribosomes, so they're the ones that read that recipe and actually put together the ingredients. Describe the genetic code and how the sequence determines the proteins, and why every cell does not express all of its genes. So hopefully that all makes sense now, and you can kind of relate it to everything that we have learned in this unit and your daily life. Like, that's how you look like you, because you're expressing your unique set of genes that make you look like you. Yeah, so that is lecture 7, molecular biology.